Hello, everyone, and welcome to the ROSA Worldwide webinar series. My name is Mike Reed. I'm the Chief Scientist at ROSA & Co., and I'd like to welcome you all to this month's webinar. During the webinar, you can ask questions of the speaker. There is a space on the control panel to the right of your screen where you can enter your questions, and we will read them at the end of the presentation for the speaker to answer a select number of questions. Our speaker today is Michael Monine. Michael is a pharmacometrician and a clinical pharmacologist at Biogen. His role is to develop PK/PD models to support preclinical and clinical development of CNS therapeutics, including ASOs and large molecules. Prior to working at Biogen, Michael worked at Novartis, where he was a member of the modeling simulation group. He received his PhD in chemical engineering from the Technion Israel Institute of Technology in Haifa, Israel, and did postdocs at Princeton, North Carolina State University, and Los Alamos National Lab. Please welcome Michael Monine. Go ahead, Michael.
Thank you very much for the introduction, Mike, and thank you for giving me this opportunity to present this ROSA webinar series. So I will talking today about prediction of PK/PD for intrathecally administered antisense oligonucleotides. So my presentation will cover briefly basic aspects of ASO distribution after intrathecal administration, some characteristics of exposure, and experimental methods, and also modeling. And we also talk about challenges associated with the intrathecal injection and the translation from preclinical to clinical studies. So what are ASOs essentially? So ASOs are short single-stranded synthetic nucleic acid and chains, typically 18 to 20 base pairs in length, and the molecular weight typically ranges between 6 and 8 kilodaltons. And ASOs are typically hydrophilic and highly water-soluble. And by design, the intention is to basically, well, the mechanism of action of ASOs is to bind specific sequences, for example, on RNA, and produce a certain signal that translates into the protein modification. Specifically, in CNS targeting ASO aspects are the following. So both size and charge for most ASOs prevents them from distribution to through blood-brain barrier. So that implies that if we administer ASOs systemically, they will not reach sites within the CNS. And therefore, ASOs must be administered directly into the CNS, for example, using intrathecal route of administration. So mechanisms of action for ASOs can be different depending on the target, and typically ASO trafficking within cells may occur through different pathways, and there are several hypotheses, including so-called productive and non-productive uptake, that both result in different outcomes. In productive pathway, ASOs molecules internalize, they go into endosomes, and then they are delivered to the nucleus, where they interact with mRNA or other genetic targets. In non-productive pathway, ASOs end up in lysosomes and essentially get degraded. Depending on the target, ASOs are designed to work through different mechanisms, and for example, so-called gapmers, they are designed to basically bind mRNA and reduce production of certain proteins that might be associated with with pathology. Another mechanism is so-called splicing modulation, as in case of Spinraza, for example, or nusinersen, and this is associated with basically rescuing a missing sequence and producing the right protein which is missing in the diseased people, in people with SMA. So basically, in this way, we are able to kind of replenish supply of SMN protein, which is downregulated in patients with SMA. So what is intrathecal administration? So first of all, the idea of IT administration is to allow the drug to bypass blood-brain barrier. In IT administration, the drug is administered into cerebral spinal fluid, CSF, which is not homogeneous, as it's slowly moving and slowly stirred fluid. And there are some mechanical factors that affect distribution within the CSF after IT administration, and those factors are heartbeat, breathing rates, and some other mechanical stresses that modulate a kind of distribution along the spinal CSF canal. Also, factors associated with the IT injection, they also modulate kind of delivery of intrathecally administered drugs. Another important aspect to mention is that right after IT administration, drugs can be drained from CSF to blood. And as shown in the figure, so IT administration site is close to famous epidural space, but the difference is that, again, as I mentioned before, so the introduction is made into the CSF canal. And as we know, CSF is produced in choroid plexus in the middle of the brain, and then it slowly moves around the cranial portion of the brain, and certain portion of it also goes to the spinal CSF canal and goes around the spinal cord, along the spinal cord and coming back. So up to date, there are quite a few intrathecal ASOs available or in development. So here is a list of ASO molecules that are currently in clinic, like nusinersen or tofersen from Biogen, and some other ASO molecules are still in development in phase 1 or up to phase 3. And they are targeting different CNS diseases depending on the, again, on the target site. So each of these molecules have specific sequence. So what we know about just overall systemic pharmacology of ASOs. So after intrathecal administration, similarly to subcutaneous route of administration, ASOs undergo rapid absorption into the systemic circulation. Of course, the intent, as I mentioned before, is to deliver ASO molecules to the CNS targets. However, a significant portion of a dose gets cleared to basically is transferred to the systemic circulation right after IT dose. However, we know from PK studies in animals and also in human that mean plasma concentrations generally decrease quite substantially by 24 hours after the injection in plasma, and it happens due to relatively fast absorption by the liver and kidneys. And distribution to other systemic organs are typically minimal. So systemic clearance of ASOs typically occurs due to metabolism or blood excretion, or due to metabolism in the liver or excretion in urine. So given the fact that ASOs are relatively clear relatively rapidly cleared from blood, there is a relatively low risk of DDI and QT prolongation. So coming back to intrathecal administration, so there are limited approaches to measure exposures in the CNS after IT administration. So biopsy and microdialysis are not always possible in clinical conditions. So typically we have a possibility to sample from CSF. However, CSF in human typically pre-dosed does not represent the target site, which is of course in the CNS tissue such as brain and spinal cord. And CSF exposures are mainly associated with residuals that are or like essentially residual concentration that is typically transferred back from tissues to the CSF, and it also so from the CSF concentration we can also infer something like elimination or terminal half-life, but not the absolute amount in the CNS. However, CSF concentrations are still informative, and I will talk about it a little bit later. There are also new applications of imaging, and some of these imaging techniques are quite promising. They typically involve labeling technology. This essentially when a molecule is targeted with a radioactive label, or it's pre-targeted with some other molecule that reacts after another molecule, a titer is injected at a certain time point, and then we are able to get intensity or radioactive signal at certain time points in vivo. However, this technique is still rather still qualitative rather than quantitative. So to characterize a distribution of ASOs, we at Biogen typically use preclinical data, and this data is coming from monkey experiments, essentially from NHP studies. So essentially, due to close similarity to human, NHP is considered as a suitable species to evaluate PK of ASOs after intrathecal administration. And when we conduct NHP studies, we typically measure concentration in lumbar section of CSF, and then we also sample tissue locations after animals get sacrificed at certain time points, and we collect spinal cord, lumbar, thoracic, and cervical regions, and also some brain regions and systemic organs such as liver and plasma. And of course, plasma can be collected during the in-life period of the study. So there are several modeling approaches to describe distribution of intrathecally administered ASOs. And first, compartmental or population PK models have been developed recently to describe some clinical data for nusinersen, for example, and tominersen and tofersen also now. And these models are based on the clinical PK data in CSF and in plasma. However, those models are mostly empirical. So they take into account individual differences, as they are a population based in nature. However, they are empirical. So more physiologically based approaches have been recently proposed, and they are mostly based again on preclinical studies, as I mentioned before. So NHP is a good source of data, and but those models have to be properly translated from monkey to human. The third approach basically relies on the computational fluid dynamics methodology, and in this approach, so we look at the dynamics in CSF after the injection, and this is a mechanistic approach, and it takes into account certain parameters such as geometry of CSF canal and characteristics of injection, such as volume and flow rate and ejection rate. So so those approaches are particularly useful when we try to optimize IT injection parameters and essentially get better delivery to certain areas along the CSF canal in the spinal cord or in the brain. So in my presentation, I will focus mainly on the PBPK or physiologically based PK model approach that we developed at Biogen, and it neglects certain mechanistic characteristics. However, it still relies on a physiological structure and connectivity between different compartments in the body. So in this schematics here, you see, first of all, in the middle, it's a 5 compartments that represent a CSF canal, and we start with the lumbar section where the IT dose is administered, and then basically the drug or ASO can distribute along the CSF up to the distal brain location, which is basically cranial portion of the CSF. And CSF can also, well, essentially the drug can also transfer from CSF to adjacent tissues, while ASO resides in the CSF canal, like in the spinal CSF canal, it can go to directly and get absorbed by the spinal cord tissues, and in the brain areas, it can be absorbed by the certain brain regions as well. And typically the nature of these regions is defined by the data. So in NHP studies, we typically collect samples from lumbar, thoracic, cervical regions of the spinal cord, and then certain regions in the brain, such as cortical areas, hippocampus, pons, cerebellum, and sometimes some midbrain structures as well. So in this model, we also describe the transfer from CSF to systemic circulation and absorption by the peripheral tissues and liver and kidneys. So this model has been developed in collaboration with Ionis, our partner, and basically the design of this model is primarily based on the nature of NHP data that we typically get from tox and by distribution studies. So this is just to illustrate what type of data we typically include in this model. So usually it's very sparse data, as such experiments include like 10 or maybe 20, 30 animals. So usually we have a very limited number of time points, and every single circle here indicates basically a sample taken from a single animal, because it's essentially it's a terminal or sacrifice study. So in tissue, every measurement comes from a single animal. However, in the CSF and in plasma, we have more frequent sampling, which is essentially done in the in-life period of every animal. Therefore, we can characterize those profiles with a little bit better precision. So given the nature of this data, we cannot really use population approach. Therefore, we describe this data using a naive pool approach, when we essentially interpret all data points in tissues coming from different animals as kind of representative of the average behavior for the whole species. So we take this model and feed the essentially feed the parameters of these models, and the parameters are mainly transfer rates that describes rates of transition from one compartment to another. And after the fitting, so we usually get agreement to the data that is kind of represented here. So it is an example for tofersen ASO, which is representative across other ASO molecules, other gap mirrors. So what we see from this data, first of all, so we can tell first that there are different time scales and the different phases of behavior of PK. So first of all, if you look at the so if you look at the lumbar CSF plot, we see a multiphasic nature of concentration decline. So first, right after IT dose, we have a peak, obviously, because lumbar CSF is a site of administration. Then over the first couple of hours, or maybe up to 1 day, we have a very rapid drop, and then we have a kind of slower elimination that actually reaches to the sort of quasi-equilibrium. And similar profile is observed for plasma concentration with a little shift in terms of the peak time. And the peak time in plasma typically occurs a few hours after IT administration. And in tissues, we typically observe more kind of smooth profile. So it does not have this sharp peaks. It's more like stepwise accumulation after each dose. And I forgot to mention that in the tissues, so here in this example, it shows basically 3 doses given every 2 weeks in tissues. So a few important conclusions we can make from this, analysis. And so first of all, if you look at the totality of the data and the model predictions, we can kind of derive the big picture of the distribution. So first of all, so this plot on the right shows this overall distribution kinetics. So first of all, we see that after initial peak in CSF, we see significantly smaller peak in plasma, but it follows the CSF quite rapidly, with a few hours delay. And then we also, well, as concentration in the CSF decreases, we also see significant, we also see absorption in the CNS tissues. So the dashed blue line indicates a sort of total amount observed in the measured CNS tissues, and also it is essentially the model fit of the data. So and as we can see from this kind of modeling and experimental assessment, that that about 4 or 5% of the dose reaches CNS tissues in about 1 day after intrathecal administration. So and the rest is something like 95%, or even more, leaves the CNS and goes to systemic circulation quite rapidly. So it may appear as a kind of significant drug loss. However, if you administer IT, well, if we administer ASOs using a systemic route, then we will not be able to reach even that amount of exposure in the CNS. So this is just to, again, quickly illustrate basic mechanisms of distribution. So if you look at the profiles in the CSF, and also in some CNS tissues, we can see that in the elimination or terminal phase, the slopes, if you look at the log scales, right? So the slopes are essentially parallel, although the absolute amount of concentrations is substantially different. So CSF concentrations do not represent concentrations at the at the CNS target sites. However, we can see that at least elimination half-life can be represented quite well by the CSF measurements. Now, so we translated this NHP preclinical model to human by applying simple scaling factors that are related to the volumes of organs of interest, for example, spinal cord or cortex in NHP and in human, typical values, and we simulated certain scenarios and compared to data available from the autopsy from the autopsy bank. Essentially, several patients in the tofersen program and also in another investigational program at Biogen passed away, and their tissues were analyzed for concentration of ASOs. And as you can see here on these plots, predicted versus observed concentrations are aligned relatively well, reasonably well, given the fact that variability is substantial. So essentially for every subject, we simulated exactly the same scenario as was basically, exactly the same dosing scenario as the subject received like certain doses of tofersen, for example, with a certain intervals between doses, and the observations and autopsy was done like at the time of death and at a certain time after the last dose. All these factors were taken into account in the modeling, so we were able to simulate. And now if you look at the agreement, so for the range of doses tested for tofersen, at least between 20 milligram and 100 milligram, we see that the agreement is reasonable over the stretch of this concentration range. In case of C9orf ASO, it's a little bit more like a cloud rather than a trend, because the doses were not significantly different. So essentially here we see a stretch between like 10 milligram up to 35 milligrams. So we are collecting and analyzing more data, and after we get the data from high doses, we will include this in the analysis as well. But so far, it provides us with some confidence that NHP, first of all, NHP is a reasonably good model, and second, linear scaling works reasonably well, at least as a first approximation. So what do we do with this? Predictions. So essentially, the idea was initially to predict concentrations in target tissues in human in order to support first-in-human study design. It essentially come up with clinical doses that are relevant for targeting specific mechanism of action, such as reduction of certain proteins, for example. So first of all, we built a NHP model, then we converted to the human model by scaling, and then we apply data from transgenic mouse, and this data typically includes IC50 or EC50 potency values in certain tissues in transgenic mouse, and these values are included in the indirect or direct model in tissues of interest, and then we get a range of doses and the associated degree of target engagement. Of course, when we make this prediction, we can only predict the associated target prediction in certain tissues of interest, but we don't know, and we may not know what the extent of clinical endpoint will be in that case. So here's just an example from two clinical programs, tofersen and CNNORF ASO. Both are ALS programs, and in both cases, the model gave us reasonably well predictions, and after we got human data, human biomarker data, so we were able to confirm that the original predictions using the model were quite good, actually. So the graphics here illustrate first the process of extracting the the potency values from the NHP model. Okay, sorry, so potencies from the transgenic mouse model, and then we combine those predictions from the predictions made for PK, and PK is based on the NHP model translated to human, and then after we do combined simulations using PK-PD model, PBPK-PD model, right? We were able to simulate clinical doses such as 100 milligram for tofersen, and in range of doses between like from 10 to 90 milligram for CNNORF ASO. And in both cases, we were able to predict, well, essentially the clinical data indicates that our original predictions in the CSF would result in something like up to 35 or 37% decrease of the protein, SOD1 protein in CSF in case of tofersen, and something around 60% in case of CNNORF ASO. So in terms of CSF biomarkers, we were able to kind of end up in the same, at least ballpark of numbers or percentage of reduction from baseline. As I mentioned before, even though we are able sometimes, or at least we have this this kind of guided NHP guided and transgenic mouse guided approach for predicting target engagement, we still don't know how this target engagement in specific areas of the brain or spinal cord translates into the clinical endpoint. In case of tofersen, the signal, so 100 milligram dose monthly administered intrathecally resulted in substantial decrease of neurofilaments, and neurofilaments are sort of biomarkers of motor neuron degeneration, and also it reflected in a quite consistent statistically significant reduction, improvement in the ALS statistically significant improvement in the ALS functional score. However, in case of CNNORF ALS ASO program, although we were able to predict the right exposures and hit the target, and also CSF biomarkers indicated the protein reduction which was consistent with the predictions, we were not able to obtain clinically meaningful signal. So that means that still this translation from the target engagement to clinical endpoint is still not understood, and as it involves many other aspects that require additional research, and essentially that means that the biology probably was not completely understood in the case of CNS nerve pathology, and there are many other factors that influence the ALS progression in that case. So here I would like to give a few more examples of how we use ASO-PK model in combination with other PD models. So for example, this piece of work was done in collaboration with Cosby. So basically, they developed a model of neurofilament trafficking in the body. So first of all, they developed a model of trafficking in healthy population. So neurofilaments are essentially molecules that are sort of building molecules that support the axon structure in like of motor neurons. And when motor neurons get degraded for some reason due to mutations or other factors, so neurofilaments get released into the CSF, and ultimately they end up in the systemic circulation. So then typically higher levels of neurofilaments in blood or in CSF are associated with increased neuronal death or degradation. So using the data from natural history studies and also from the clinical program, so we were able to kind of design the model, and also to introduce the treatment effect due to ASO administration. So, for example, in case of nusinersen Spinraza, right? So we introduced the drug at a certain frequency with a certain dose level, and that was direct impact into the neurofilament model, and the similar was done for the SOD1 or tofersen model. Sorry, and in that case also we simulated PK profiles using the PBPK model, and then these profiles were used as input to the neurofilament model, and essentially in that case so tofersen was included as a sort of inhibitor for neurofilament release empirically, because we do not really have this cascade of reactions of synthesis. So we still describe this process empirically, but at the same time we have a reasonably well description of neurofilament distribution overall. So here are just a few examples of the model prediction and the data. So for tofersen, that so these simulations were conducted by our colleagues from Cosby, mainly by Alessio Paris, and so in the example on the left, first they described the data from pre-symptomatic subjects who essentially have not developed the signs of ALS yet, but the neurofilament levels already started increasing. So first we describe this kind of behavior using the neurofilament model, and then we introduce the treatment in this in the same way or in different subjects, and this treatment was so the model was calibrated based on the treatment data for tofersen, And in that case, we essentially were able to describe the decrease associated with the introduction of tofersen on a monthly basis. And then in combination, essentially using the pre-symptomatic phase, and also when the disease develops and the neurofilament and the neurofilament level increases dramatically or significantly, we start treatment, and then at that, due to the model, essentially due to the administration of tofersen, we were able to kind of bring the concentrations of neurofilament back to the baseline range, as shown on this in these two examples in plots on the right at the bottom. So basically, the simulation shows the full transition starting from the pre-symptomatic phase when we have a relatively low concentration of neurofilaments. Then we approach when essentially we approach a situation when the participants start basically they emerge the disease manifestation, and then after the treatment initiation, so we're able to describe the decrease in neurofilaments. So with these examples, I came to basically conclusions of my presentation. So I would like to say that intrathecal administration of ASOs has essentially became an efficient method for targeting neurodegenerative and the neuromuscular disorders. Those projections for IT administration in humans require accurate estimation of exposures at target sites. However, it's still, accuracy of this prediction is still kind of is far from ideal. So we are still working with preclinical models and trying to project them to human situation with a lot of caveats. So and NHP model is a quite helpful tool in this regard. So and there are still high risks associated with this kind of with this approach. So first of all, it's a substantial or significant variability in PK. Then it's of course huge variability in PD effect, which is associated with the way how we access the target. Even within the tissue, the target is typically located intracellularly. So the ASO molecules, they have to kind of must reach the nucleus, and there are also a lot of uncertainties associated with the translation from the target engagement to the clinical endpoint, so which is the whole biological, the set of biological pathways that need to be studied carefully. And integrated approach with neurofilament dynamics helped us at least understand at least the overall dynamics and the modulation of neurofilament levels by the treatment. And this is another example of how this model can be used as a prognostic tool in case of pre-symptomatic development, and then when the participants become sick and develops signs, we essentially able to simulate at least for certain biomarkers such as neurofilaments. Yeah, the model can be quite useful in terms of prognosis. So with that, I would like to acknowledge my colleagues from Biogen, Ionis, and COSB. So COSB is an institute in Italy, and they predominantly worked on the neurofilament model, while Biogen and Ionis groups are mostly working on the, of course, on the directly IT, ASOs, PK, and PD aspects. So please ask questions. So I will be happy to at least try to answer some of your questions. Thank you for your attention.
Great, thank you, Michael. That was a wonderful presentation. There were a couple of questions. Actually, there were several questions from the audience. First one is, how did you scale flow rates from non-human primates to humans? So, for example, CSF draining rates and tissue uptake and release rates. Michael, can you hear me? Michael, can you hear me? Michael, can you hear me?
Unfortunately, I don't hear questions. I don't hear anything. Yeah, Mike, I cannot hear anything. Sorry. I think. Hello, can you hear me?
Hello, Michael, can you hear me?
Someone is typing that that you can hear me, but I am not able to hear anyone.
Michael, can you hear me?
Hello.
Hello, Michael, can you hear me? All right, well, thank you, everyone. We're having audio issues, so the speaker will be happy to answer your questions offline. Thank you for joining us, and please join us next month for the ROSA webinar series, where our speakers will be Colleen Witt and Bobby Sheehan, speaking about an Alzheimer's disease QSP model. Thank you, everybody, and we will see you next month. Bye-bye.